Hello everyone, welcome to another video of biology concepts. And in this video we will study the structure of chromosomes. So first of all you should know what is the difference between chromatin and chromosome. You can see this is a chromatin and this is a chromosome. Chromatin is thin. It appears during interphase before the cell divides. So it is in uncondensed form and it is almost invisible. But after the DNA duplication, like this, DNA duplicates after the replication of DNA. When the cell enters in division, then the chromosome becomes condensed, they become thick, thread-like structure, and they are almost visible. So these are known as chromosomes. Here is the chromosome, and this chromosome have two strands which are known as chromatids, and each chromatid consists of DNA molecules which are highly coiled along with the histone proteins. So you can say that chromatid have the DNA, it have the genes which express different traits like hair color, eye color, etc. And both these chromatids are attached with each other at a point known as centromere or primary constriction. So if the chromosomes are attached at the central point, then they are called as centromere or primary constriction. But if the chromosomes are attached with each other than the centromere, then this is called as secondary constriction or nuclear organizer region. So you can see here the chromosomes are constricted, and this is known as nuclear organizer region because this region gives rise to nucleoli during interphase before cell division. So that's why it is called as nuclear organizer region. And besides secondary constriction, you can see that these ends of the chromosome become knob-like structure. So they are known as satellite. And this region has useless sequence of DNA. So it is called junk DNA. Junk DNA is the useless sequence of DNA. And the terminal ends of chromosome, these terminal ends of chromosome are known as telomere, and the function of the telomere is to prevent the two chromosomes to attach with each other from their ends. So this is telomere. In this diagram you can see different types of chromosomes on the base of position of centromere along the length. So you can see this is known as metacentric, because here the centromere is located exactly at center. So this is known as metacentric chromosome. And if the centromere is located slightly away from the center, if it is located slightly away from the center, then this is known as submetacentric. And if the centromere is located near the ends, then this is known as acrocentric. And if the centromeres are located at the ends, like here, so this is known as telocentric. So these are the types of chromosome on the base of position of centromere along the length. In this diagram, you can see the DNA having the width of 2 nanometer is coiled around the histone proteins. Why? Why the DNA is coiled around the histone protein? DNA is negatively charged because of phosphate group in it, and histone proteins are positively charged because of positively charged basic amino acids in it, which are lysine and arginine. So negatively charged DNA has strong affinity toward positively charged histone proteins. So here the DNA, the 200 nucleotides of every duplex DNA is coiled twice. It is coiled twice around histone proteins, and this is known as nucleosome. And here one histone molecule, this one histone molecule is attached to the segment of DNA which is known as linker DNA, and it is attached between every two nucleosomes. It is present here, then here. So it is attached between every two nucleosomes. And in this way, the whole DNA is turned to be like a string of beads. because here the string is the long length of DNA and the beads are the histone proteins. So this is also known as nucleosome string. And now this nucleosome string begin to coil again about its axis and form a thick chromatin fiber of almost 30 nanometers. And this chromatin fiber found in G1 and G2 phases in interphase, which is before cell division, and this chromatin fiber shows two kinds of regions. One is euchromatin and the other is heterochromatin. Heterochromatin region is highly condensed region, but it is unexpressed, and euchromatin is uncondensed region, but it is expressed. But when the cell undergoes division, euchromatin, which is uncondensed, became condensed so that a uniform chromatin fiber is formed here. And now the cell division begins, where the chromatin fiber coiling gives rise to supercoil and form a thick chromatin fiber of diameter 300 nanometers here. And this supercoiling establishes the chromatid of almost 700 nanometers because of further coiling of DNA around itself. And then here the chromosomes of almost 1400 nanometers appeared. That's all about the structure and composition of chromosomes. I hope you like this video. Please like and subscribe this channel.